Hopefully it will. Wonderful. Thank you everyone for joining. This is a really exciting topic and great speaker. So thanks to Linden, maybe I should, Dr. Parkes, I guess.
Linden is fine.
Brief intro. Dr. Parkes is a postdoctoral researcher in the Department of Bioengineering at the University of Pennsylvania. Linden is a computational neuroscientist working on understanding the biological basis of psychiatric disorders, uses the MRI, and draws on tools from network science and data science to build statistical models that forecast the emergence of psychopathology through development. Very fascinating topics. Linden will talk about his work conducted during his PhD, examining the efficacy of motion correction methods for resting state fMRI data, with a particular focus on understanding the implication for case control studies in psychiatry. So without further ado, Dr. Parkes, thanks again for taking the time. Please take it away.
Sure. Well, thank you very much for having me. Let me first see if I can share my screen. Okay, can you see the presenter view or the slide view appropriately?
Slide view, I think.
Cool. All right. Thank you very much, Pradeep, for having me. I'm glad to be here. It's a privilege to be invited to talk about this work. This was a lot of fun to do this paper. It's a little old now, but hopefully it still has something to add of value for the group here today. I just also want to emphasize that I'm very happy to be interrupted and sort of make this very kind of discussion based. We can sort of pause at whatever time people want to and ask questions. I've got the chat up here, but Pradeep, perhaps if I lose track of it, perhaps you can stop me if you think it's necessary. But very keen to have lots of discussion. There's lots of people have contributed to this space, not just me, and I'm sure there'll be and there are lots more people thinking about it ongoing. All right, so today I'm going to talk.
Sorry, do you need to run right after 12, or would you have some time after 12?
Yeah, no, I've got time. I can stay after 12. That's totally fine.
Yeah, usually these things run for 2 hours because there's always a lot of questions and whatnot, but feel free to leave anytime. Everything is being recorded. You don't need to screenshot or whatnot. We'll share the video as soon as possible. So those who can stay around, ask questions, let's get to it, and try to keep a QA angle in mind. That's the only request I try to make, because the overall goal is how do we improve the quality of the results, not just the input data, but also the results we publish and report. Yeah, thank you so much.
Sure. So today I'm going to talk about this paper that we titled, it's a bit of a mouthful, an evaluation of the efficacy, reliability, and sensitivity of motion correction strategies for resting state functional MRI. This is work I did back during my PhD and published it in 2018 with my PhD advisors, Ben, Murat, and Alex. Now, as Pradeep sort of asked me to make this quite didactic, so I'm going to include some high level sort of educational stuff, which will probably be very common knowledge to most of the audience, but hopefully it gives a little context, especially for the way I kind of think about analyzing the data. So the first thing is we've all probably heard of the echoplanar image. So a little primer on our assumption about what this data is doing and what it allows us to understand about the brain. So the echoplanar image is, of course, sensitive to this blood oxygen level dependent contrast. And the assumption being that as neuronal firing goes up in some part of the brain tissue, some region of the brain, the amount of that tissue extracts oxygen from the oxygenated blood, That increases deoxygenated blood, right? The oxygen goes down. And the EPI is sensitive to this change, this ratio of change between oxygenated and deoxygenated blood. And so as this ratio is changing throughout the brain, we think that this is signaling variation in brain activity. This is kind of our assumption of causal change between neuronal firing and what we get from the EPI. And of course, what we do in the MRI is we get a whole bunch of these images, typically at some, some temporal resolution of maybe sub-second, but typically sort of 1 to 2 seconds for standard acquisitions. We string them together, and then that way we can sort of sample a region or a voxel from the brain. And if we have this EPI contrast, we kind of say, well, this bold time series is our changing brain activity over time. Now, I come at this from a network neuroscientist perspective. So I typically care about building whole brain functional connectomes and analyzing the topology of the network. So what I tend to do is, I will sort of break the brain down into a series of regions, say a couple of regions here, region 1 and region 2, and get that bold time series out from both. And then simply say, well, if there's a statistical dependency between those two time series, say they're correlated at 0.7, we're assuming a functional connection is happening as a function of this just temporal dependency between those bold time series. Then we do this at the whole brain level to sort of populate our adjacency matrix one at a time with all of these correlation values until we have a whole brain connectome that describes how the brain regions in the brain as nodes are connected by a series of edges. And then someone like myself comes along and wants to ask questions about how connectivity changes in the brain between people, how higher order aspects of topology vary, and topology being just the idea that the relationship amongst these nodes, the actual pattern of connections is in itself important. But there's a whole lot between those two steps, right? If we start at EPI on the far left, some raw data straight off the scanner, getting to a functional connectivity matrix on the right, there's a whole host of stuff that goes on between those 2 points. And this broad, this whole space of pre-processing has its own sort of garden of forking paths. There are heaps of steps involved in pre-processing to go from raw data to a functional connectivity matrix. And so, and the choices we make as scientists has a big impact on that output matrix on the right-hand side here. And so what I'm going to do today is I'm going to sort of just zero in on one of these steps and talk about how in this work we, which is motion correction in this case, I'm going to talk about how during my PhD, myself and Alex were kind of very interested in understanding the impact of making these decisions and the impact of different approaches that you might take to outcomes that we cared about. So first, the big issue. So this will be common knowledge, I'm sure, to everyone here, but people don't tend to sit still in the scanner. Even people who feel like they are move in very subtle ways, and those subtle movements can have big impacts on that bold contrast on that time series of activity. So what I'm just showing here is that typically what we do, this is just a readout from common software package, statistical parametric mapping, which summarizes a single person's movement in the scanner over time, over a few hundred volumes, a few hundred EPIs. And it summarizes them according to 3 displacements in translation, so they can move around in the X, Y, and Z in 3-dimensional space. And then they can also, pitch, roll, and yaw, right? They can rotate their head around several axes. And then so when we scan people, we can get this information out and we get some sense of how much they move. And we know now through a long history of research that even subtle movements, even sub-millimeter movements can be quite problematic. Now, historically, this movement has been I guess less problematic for task-based designs. Not that it's not problematic at all, but tasks have the beautiful privilege of boosting signal to noise ratio, making these subtle movements, the effects on the time series less apparent. But in resting state data, we have no such privilege, right? We just have essentially one big long trial with a relatively low signal to noise ratio, which means this effect of people moving around in the scanner has some serious problems. And so around about I want to say about 10 years ago now, the field got caught up in a huge panic about how, the way we would kind of, the standard way in which we would control for motion, that we sort of historically inherited from task-based designs wasn't kind of working as advertised for the resting state data. And this was probably most famously popularized by Jonathan Power, who did a series of studies on this, but lots of people have worked in this space. This is a figure from one of Jonathan's earlier papers showing that the top plot is showing the percent change in the bold signal over time. And then the bottom plot is showing, and it's showing that as a series of compartments in the brain. And the bottom plot is showing just a single trace of what's called the frame-wise displacement in millimeters, which is just a collapsing of those 6 parameters I showed before, and just summarizes kind of just in general how much a person moved between 2 frames. And you don't even need to do statistics on this to see the issue. It's quite apparent to the naked eye, right? You can see that sort of big changes in movement or big periods of large movements in the scan are time locked to relatively large changes in the bold signal. And importantly, this is the top plot here is the change in bold signal after kind of regressing out those 6 motion parameters, showing that this problem lingers a great deal, even if we try to sort of clean it with historically standard ways. And then the other thing that makes it very easy to see is, again, popularized by Jonathan Power, this idea of a carpet plot. So again, this is from his work and Neuroimage in 2015. What we're showing here is along the rows of these black and white plots, voxels, or they could be brain regions, and then time is along the y-axis, and activity has been just projected as like a heat map in terms of black and white signals. And you can see that before motion correction, there are these, where we see these large movements that the participants are eliciting, we see these very large synchronous changes in the bold activity. But again, importantly, after we do motion correction, it's very easy to see, even with the naked eye, that those synchronous changes across the brain are still very much present. And of course, given that I mentioned before that functional connectivity is defined typically with a linear statistical dependence between the time series, any kind of big global shifts like this, even like relatively subtle ones after denoising, will drive up the statistical dependencies between our time series and sort of give the appearance of connectivity or functional connectivity where there may very well be none. And then, of course, this problem of motion-induced spurious connectivity is compounded in many of the analyses we might do as scientists, particularly, for myself, those interested in studying individual differences or studying group contrasts in patient groups. This problem of contamination gets exacerbated because we know that patients often move more than healthy controls, even patients who don't have movement disorders, but psychiatric disorders that wouldn't classically be considered movement related still tend to move more than healthy controls. And also, from a developmental perspective, children tend to move more than adults. And so, if you have, this is an old plot from Ted Satterthwaite, again, showing very clearly between the ages of sort of 8 and 22, the amount of movement on average that kids engage in the scanner goes down as a function of age. So this provides a huge problem, right? Because if you want to compare young versus old, patients versus controls, and one of those groups moves more and therefore has more contamination, then you end up with this a great deal of difficulty teasing apart whether or not what you're seeing in the data is because of some sort of pathophysiological signature, or whether it's just that one group is moving more or less than the other. Move along slides, let's go. Okay, so in response to this huge problem and in response to this panic in the last sort of 10 years or so, the field has proliferated an enormous number of methods to deal with this problem. And today we're going to talk a bit more in detail about some of them, but certainly not all. This is just a few examples of the, of those methods. So we can do head motion parameter regression, which is what I've already talked about. But then there's all types of other methods like volume censoring, methods based on principal component analysis, methods based on independent components analysis, global signal regression, and many, many more. And so, you know, during my PhD, my initial response to this was just to be overwhelmed. I didn't really know exactly what to do, which to use. It wasn't quite clear to me back in 2015, 16, when I was trying to do these analyses, which ones to use. And so, and in addition to that, in around about that time, around 2015 or so, I was doing an analysis with one method. It was just one of these denoising methods. It was a PCA-based method that you know, I was just told to use, right? There was a postdoc in the lab at the time who said, just use this one. And that's what I was doing. And then it was around that time that a whole new method came out from Christian Beckman's lab, which was based upon independent components analysis. It was called ICA Aroma. And Alex, you know, in one of my supervision meetings just said, oh, just double check that your results are robust, right? I was doing some case control comparisons with OCD. And so I dutifully went and reran those analyses like I should. And I found that almost nothing was the same, right? So comparing just whatever I was examining at the time, comparing one PCA-based method to an ICA-based method, just from the literature, they gave very different results. And so that caused Alex and I to panic, and that's what motivated us to do this study. So what we set out to do was to compare the efficacy of what was available at the time for motion correction methods. And we were interested in two things. We were interested in, you know, we wanted to understand the effect of these different methods on, one, the sort of residual relationship between in-scanner motion and resting state functional connectivity, right? So what contamination is left over effectively. And as I said before, we were very interested in, well, what is the impact of these decisions on case control studies in psychiatry? Because that was the focus of my PhD. And as I mentioned, you know, comparing two different methods arbitrarily got us a very different set of results. And so we kind of embarked on this idea of trying to figure out which one we should be using. So before I talk more in detail about this paper, I just want to take a quick note to say that this work that we did really was a contribution among many. We are by far not, you know, we are by no means the only people to be working in this space, and we're by no means the only people working it before we did it. So this paper, my paper, sort of fits into a long history of this type of stuff, dating back more than 10 years. This is just a list of a handful of the papers, other contributions from people at various scales, and there are many, many more. So I apologize if your favorite study isn't on here, but I just want to make it very clear that we are not lone wolves in this space by any means. And so what we set about to do was to create a fairly comprehensive list of about 19 or so motion correction pipelines that represented different various combinations of the available methods that we thought were sort of a good representation of what people were doing in the literature at the time. And so now what I'm going to do is, in an effort to make this didactic, I'm going to kind of step through just at a high level, a few of the key components to these pipelines, give people some intuition. Again, I'm sure a lot of this will be well understood. And then we'll sort of come back to this slide and talk about the results. So of course, the first way that people do it, I've already mentioned, is just head motion parameter regression, right? And so typically in scan acquisitions, we don't actually actively acquire data about participant motion while they're in the scanner. Instead, what we typically do is we kind of have to derive it algorithmically from estimates of how much the EPI data has actually been displaced frame to frame. And so, you know, what happens is that we see on the left, typically when we acquire the data, if you look at the EPI frames over time, you can quite clearly see that the brain has moved around within the bounding box of the MRI. And then so a very simple pre-processing method, of course, is to just spatially realign those. And then as we do that spatial realignment and make sure the brain is in a sort of fixed common space across time, at least for the subject space, that's what gives us out these 6 motion parameters, which we've talked about before. So the kind of common way that people were doing this at the time to do head motion regression was they would take those 6 parameters and then they would expand them to try and account for sort of 1 frame delays on the effect of the bold signal. So do things like take the temporal derivatives, which is just to sort of take the motion parameter from one time point and subtract it from the previous one. So that takes you 6 parameters to 12. You know, you've got 6 normal and 6 temporal derivatives. And then on top of that, they would include quadratic terms in the nuisance regression design matrix by effectively squaring those 12 traces. And the idea of including the quadratic term in the design matrix was to remove you know, the impact of the motion that was maybe potentially non-linear on the bold data. So that's the basic kind of head motion parameter regression. From there, another very popular technique is to look for sources of noise, motion and otherwise, in the non-neuronal brain compartments, right? So the kind of classic way of doing this is to pull out the average signal from the white matter compartment in the brain and the average signal from the CSF compartment in the brain. And then the assumption being that if we have those two signals, those two bold signals, if those signals share variance with the signal variance that we see in the gray matter, well, that shared variance should presumably be noise because those compartments are non-neuronal. So whatever is shared between them and the gray matter, in the gray matter, that must be noise. So the classic kind of way is to take the mean 2 signals of those compartments and regress them out of the signal or put them into your design matrix. But there are slightly more expanded ways of doing this based upon principal component correction, which is on the right. Very simply, the idea here is instead of just taking the mean signal in each compartment, we do a PCA analysis and retain some of the principal components or some number of the top principal components within each of those compartments. And the idea being there is that you get a more diverse representation of whatever non-neuronal signal is housed in those compartments. The next class of methods we examined was those based upon independent component analysis. Independent component analysis is based on the idea that you try and extract a set of sort of independent spatial components from the brain data, and then you have to make decisions. And now this is, you know, effectively the whole brain data as opposed to just the white matter and CSF compartments. And then you have to make decisions about whether or not those spatial components are noise or neuronal. And then once you've made a decision about the components that are noise, you take the time series associated with those components and you add them into your nuisance regression matrix as well. So this picture on the left is pinched from the website from fMRI Prep. This is the output from what's called ICA aroma. So you can see here, these brains represent the spatial components from the independent component analysis. And then the time series, the top time series associated with each one is represented with the actual time series of the component. And then the bottom one is the power spectrum of that time series. And then, but, you know, either way, you know, irrespective of, you know, excuse me, once you get these components out, you have to label them, right? That's what I mentioned before. And there are multiple ways of labeling them. So this on the left here is based upon an automatic labeling approach called ICA aroma, which is by Preprocessing et al. From 2015. And ICA aroma basically just uses a very simple classifier to try and determine which components are motion related based upon a few simple criteria for example, the idea that you know, say more than 10% of the component voxels are within CSF, for example, would be a criteria. And you can read the paper if you're interested in more. But the general idea was, you know, instead of going through and manually labeling which components are noise and motion, which is very laborious, they defined a little simple classifier to do this automatically for people. And then there are other more elaborate approaches called ICA fix. This is very popular amongst the HCP data, for example, which involves sort of manually labeling the components and then training a classifier based on that. That has the advantage of being better equipped to deal with different types of contamination, not just motion. So it does particularly well with the types of artifacts you see specific to multiband data. But of course, it's much more laborious and takes a lot more time and effort on the researcher's behalf. So either way, once you've decided upon your labeling, you know, for example, here, on the left, the components in green have been designated as signal and the components in red have been designated as noise based upon the aroma algorithm. And this is provided with fMRI Prep. Once you've made that decision, then again, you've got some noise components and into the noise matrix they go.
Sorry to interrupt. Can I ask
Go
a
ahead.
quick question? Yeah.
Yeah.
Can you go back back a slide? Yeah.
Mm-hmm.
So what give us a few examples of non-motion components that require manual identification from an expert rater for ICFX?
Oh, I see. I don't have any handy on the slide here, but this is not something I've got deep expertise in because I don't tend to work with the data so much. But some of my colleagues, for example, who have, you often see in the multiband data, you can see specific kind of striping artifacts that are apparently linked to the multiband acquisition specifically. And so if you were to use, say, ICA aroma, which has a simple classifier designed to tune to pull out motion, then the concern with that is that you would miss those components, right? Those components would be erroneously labeled as signal and then not removed.
Thanks.
Okay, last couple of sort of classes of approaches that we tried was to look at volume centering and global signal regression. So volume centering comes in a few different flavors, but the general idea is that instead of trying to fix the whole time series through a nuisance regressor that might be the motion parameters or something from independent component analysis. The idea is to sort of zero in on the volumes that are probably highly contaminated by motion, and then either delete them or interpolate over them, or through something like a spike regressor, try to sort of squish the motion contamination in specific volumes. And this is a method that is particularly championed by Jonathan Power. So he has a whole string of papers where he sort of develops this approach, and he's a big proponent of it. And then the second one on this slide is global signal regression, which is every fMRI researcher's favorite curse word, and it's every reviewer's favorite review point, is to check the results with and without GSR. It's a particularly controversial a method. It basically involves taking a single signal averaged across the entire brain compartment. So it involves the white matter, the CSF, the gray matter, etc., and then taking that global brain signal and regressing it out of the data. And then your analysis is effectively asking, what variance in the data, what you're then examining the variance in the data after you remove that big, broad global signal. This has been shown to be quite effective. It's because it's quite I guess for lack of a better word, an aggressive technique, but it has been shown to be quite effective at minimizing these big time-locked bands of activity that we see in these carpet plots. But it remains one of those challenging things where lots of people are debating to what extent it removes too much signal. And so it remains something that people kind of do their analysis with and without, as far as I can tell. So after that kind of high level thing, we returned to this set of pipelines we used. So these pipelines for us kind of group broadly into 5 different categories, whether they're kind of based upon a fairly standard combination of head motion parameters and just mean time series from the white matter and CSF compartments. So that's the sort of first 6 pipelines here. They just varying degrees of expanding upon those nuisance regressors using those derivative terms. Then the third lot uses PCA. So this is a method called anatomical comp-core. This is the method I started with in that anecdote I gave you early on. And so we have a whole bunch of pipelines that sort of revolve around using anatomical comp-core in a few different permutations. Then the 4th batch is pipelines revolving around that approach ICA aroma, which I showed you before. And then the final batch involved sort of augmenting the sort of standard head motion parameter and white matter CSF pipelines with some version of scrubbing. And we have a few different permutations of that. So, the goal of the paper was to examine the efficacy of these different pipelines, see to what extent motion contamination was effectively removed or not, and also to examine how the results we care about, so case control effects in psychiatry, vary as a function of these pipelines. So how did we benchmark them? So we benchmarked them in two broad ways. There are a bunch more things that we did in the paper, but I just want to focus on a couple things we did here today. The primary one is this idea of QCFC correlations. The simple idea here is that for every subject, you take their frame-wise displacement, that single trace of how much they moved frame to frame, and you average it over time to get a single number that represents just how wriggly the person was in the scan. A higher number, they moved more. And then so for every subject, you have one value that represents their degree, the degree to which they moved. And then once you have every subject's functional connectome, you can go through every edge in that functional connectome. You can pull out the functional connectivity estimate across subjects for that edge, and then you can correlate it with the subject's measures of that movement, of the mean FD. And that's what I'm showing here. So every value in this vector represents a dummy subject. And then, so we have their frame-wise displacement and their functional connectivity for a specific edge. We do that across all edges in the brain, and we build up a distribution of correlations between, cross-subject correlations between motion and functional connectivity. And then the second thing that we're interested in is this idea of distance dependence. So what I'm showing here just as an example, of course, intuitively, we understand that regions are spatially embedded within the brain, and therefore the connections formed between regions occur across sort of different physical lengths, right? So there'll be connections at a long range, connections at a short range. Even though they're functional, they're sort of positioned in different places, so they have a distance. And the literature before us showed that there's a sort of strong distance dependence on this contamination with respect to motion. And so what we did was, as well as other people have done, we took those correlations, those QCFC correlations on the y-axis, and plotted them against distance between the regions that made up those connections. And the idea is we can see that contamination was shorter, well, contamination was higher over shorter ranges and tended to decay over longer range connections. So in either case, so from there, all right, so on to the results. So what I'm showing first here is the QCFC correlations in this plot here on the left. What we do is we summarize those distributions of correlations as the proportion of significant correlations, uncorrected. We use uncorrected here because this is one of those, I guess, rare circumstances where correcting for multiple comparisons is the sort of less stringent thing to do, because of course we're interested in if there are lots of edges in the brain that are significantly correlated to motion, if we correct for multiple comparisons and we remove all of that, well, then effectively we can drive our benchmarks down to zero, and we can make the results look really good by just overcorrecting. And so what we do is we use uncorrected significant results here, and then we plot the proportion of significant motion associations across these pipelines. So the main take home here on this first left plot is that these kind of classical expanded head motion parameter or non-expanded head motion parameter pipelines aren't doing so well in this data set that we had from Beijing. And in the worst case scenario, if you're only using head motion parameters, more than half of your connections are still showing significant associations with motion. And you can see that global signal regression massively reduces that. But we also find that there's many of the other pipelines do fairly well. The differences are fairly subtle, and with there's always a slight gain with GSR, but generally, as long as you're using some of these, I guess for lack of a better word, more advanced approaches, you're doing reasonably well. And then on the right hand side, we're showing just the full distribution, so you can just see the full measure of central tendency if you're interested. And so we thought, well, that's not super informative for us. And so what we wanted to do, and what we found is with this Beijing data set, the motion is actually very low. So this is a data set where it just happens by virtue of probably good acquisition practices, that the general level of motion in this participant's data set is actually quite low. And so then we sort of went to replicate these analyses in another open access data set for which had higher degrees of motion on average. And that's where we start to see, we start to appreciate differences between pipelines more. And so, I don't want to go through all of this in great detail, but the main, I think, takeaways here that can be observed is that for us, in our hands at least, the ICA aroma-based pipelines were doing a bit better than the anatomical-based, the PCA-based pipelines, and then the scrubbing-based pipelines were doing better than the ICA aroma-based pipelines. One thing I want to highlight, though, and I will come back to this later, is that these numbers here in parentheses, 90%, 72%, etc. These represent the fraction of the data set that was retained for analysis after we did the denoising. And one of the challenges with scrubbing is that if you have a very high motion, or volume censoring in general, is that if you have a very high motion subject and you end up, censoring out lots of volumes, you end up with a very small amount of data left that's usable for analysis. And so, previous to us, people had made some fairly kind of rule of thumb recommendations that you needed a certain number of minutes of good, clean data to get good, reliable functional connectivity estimates. Of course, the jury is still very much out on exactly how much data and is needed. But what we did here is we took the rule of thumb from Jonathan Power and we discarded people who had less than 4 minutes of usable data after volume censoring. And that's why we have these percentages in parentheses. Are these the amount of people we retain after we do that? But there's a bit of a circular issue here because, of course, the more you're throwing out high motion subjects, the lower on average the lower motion your sample is exhibiting, right? You're basically more and more retaining this lower and lower motion subjects, the higher and higher quality data. And so there's a confound here between, to what extent are you getting good performance from really effective denoising versus good performance through just throwing out bad data. So I suspect that a lot of why these methods are, or part of why these methods come out the best is because we are throwing out some fraction of sort of bad or noisy data. Is there any questions on those plots? I know there's quite a lot to look at. If not, I'll move on.
If no one else has a question, I have a question actually.
Sure, go ahead.
I think I missed that part about the difference between anatomical CompCore and the ICA one, Is it
sorry.
just the, go ahead. Can you explain the anatomical part of that?
Sorry, yes, I fumbled my words, I missed the link. So the anatomical CompCore is the principal component-based version that I mentioned earlier. So it's the idea that you take PCA analysis from these non-neuronal compartments, right? The white matter and the CSF. And then you have a series of regressors. So you might have 10 for the white matter and 5 for the CSF, for example. And those regressors are anatomical CompCore. And then, there are various ways in which you can retain the PCA or the PCs. You can do arbitrary things by just like, retaining 5 for white matter and 5 for CSF, or you can do, enough PCs to explain 50% of the variance in the white matter and enough PCs to explain 50% of the variance in the CSF. And so there are a few different ways you can make decisions about retaining the PCs, and that's why we have several repetitions of anatomical CompCore. But broadly, that's the PCA-based approach that we chose. And then ICA aroma is the independent component analysis one.
I have a question.
Sure.
Did you do any with scrubbing but no GSR?
It's a great question. No, we didn't. The reason for that was because we were trying in this paper to reproduce how we felt people were using the pipelines we were comparing. And so in our read of the literature at the time, the spike regression approach and the different ways of doing scrubbing that Jonathan Power was discussing all included GSR, and so we kept with that.
Yeah, there was a paper by people in my group with Hangjun Zhou did this in 2013 that they looked at this kind of thing. I'll put a link for it.
Please do.
Which and they did a similar kind of set of studies, and in the end, scrubbing or censoring, whatever you want to call it, was better than including GSR. And it was able to handle these long distance correlation problems.
Yeah, cool. Please, yeah, link me to the paper. I'd be pleased to read it. Okay. So the next main benchmark is of the QCFC distance dependence. So what we're showing here on the far left is the Spearman correlation that summarizes the distance dependence effect that I described. And you can see that for the most part, they're all negative, so showing this idea that the contamination sort of drops off as a function of increasing distance. And what I'm highlighting here is just something that is a commonly reported phenomena as well, which is that even though GSR is quite good at minimizing motion just in general, or the contamination in general, it can have the effect in some instances of exacerbating that distance dependence. So you get a bit of a trade-off there where you have an effective denoising methods on one hand, but you kind of change the spatial distribution of the contamination, which gives you this more pronounced distance dependence effect. So on the top right hand corner, you can see that with one of the more basic head motion parameter and physiological pipelines, where the distance dependence gets clearly quite a lot stronger when you add GSR. And then we have a similar but more subtle effect when you append GSR to ICA aroma. And in fact, it's here there's, you would argue, there's a bit of a non-linearity being sort of strengthened with ICA aroma. So, the Spearman correlation is probably not super well suited to be sensitive to that. But just to make the point that GSR confers a bit of a trade-off. All right, so the other thing is it's not all about performance. There's this big question of there's also a question of trade-off and cost. So one of the things we did in the paper was we sort of proxied this idea of nuisance regression cost based upon a simple estimate of how much temporal degrees of freedom is lost in the data by using a given motion correction approach. And so here we estimate temporal degrees of freedom lost just very simply by virtue of using the number of nuisance regressors in the model. And so, in some of those cases, some pipelines have a variable amount of nuisance regressors across subjects. And so those pipelines have got a standard deviation bar on these graphs here, just to show how much the number of nuisance regressors varied across participants. But one of the things you can see is that one of the take homes here is across both of the data sets we looked at, things like anatomical comp-core, so the PCA-based approaches have amongst the sort of largest number of nuisance regressors in order to achieve their performance. And scrubbing comes second to that, but still has a considerable amount. And we found that in our hands, at least in the data we were looking at, ICA aroma had a relatively sort of low number of nuisance regressors, at least compared to the other good performing pipelines like volume censoring and anatomical comp-core. And so, our read on this was at the time is that we wrote in the paper is that ICA aroma was maybe a nice balance between reasonably effective nuisance correction at a sort of relatively bearable cost. So moving on to the stuff that I was particularly interested in, this is the focus of my research was at the time case control studies in psychiatry. And so what we did was we took a sample of healthy controls, about 120 healthy controls and 50 schizophrenia patients. This was an open access data from UCLA. And we just asked a simple question of, well, if we do a fairly standard analysis, like using something like the network-based statistic to compare just whole brain differences in patterns of connectivity between schizophrenia patients and healthy controls, how does that simple T contrast or two-way T contrast vary as a function of these pipelines? So the first thing I'm showing you on the left is just the number of significant connections that were sort of different in one direction or another. And we can see the first take home here is that GSR were the only pipelines with GSR showed significant effects in our analysis. But more importantly is this plot on the right. So this plot on the right is the subset of our pipelines that showed at least some significant effects from the left there. And then we just did a very simple thing where we just took out, there's only one component, only one network-based statistic component that showed differences between our patients and controls. And so we just asked, well, to what extent does that component of connections that are different, to what extent does that spatially overlap between these different pipelines? And so that's what's being shown here on the right-hand side. Higher values in this heat map show that the components were identical. These components of connectivity changes were identical across the pipelines. And then as it approaches zero, they become more and more unique. And as you can see that in general, the overlap's pretty low. In fact, the only one to show reasonably high overlap, for example, here is just ICA aroma with subtle variations to how GSR is implemented, but it's fundamentally a similar pipeline. But for the most part, when you're comparing, especially across different classes of pipelines, so across like ICA aroma and the PCA-based ones, the overlap is quite low, suggesting that when you use these different approaches, you get very different answers about that question of connectivity changes with respect to schizophrenia. And you can see this quite easily with the naked eye. So this is just sampling a couple of the pipelines. So up the top, we're showing the anatomical CompCore pipeline, the PCA-based one. And down the bottom, we're showing the ICA aroma-based one. And on the left, we're showing the contrast in one direction, so like healthy controls greater than schizophrenia, for example. And then on the right-hand side, we're just showing the exact opposite of that contrast, so schizophrenia greater than healthy controls. And then in the middle of these two plots, we're just showing, where these significant edges were, how they fell into different just canonical systems of the brain according to the Gordon Atlas. But you don't even really need to look at that high level. You can sort of, even if you just compare, the most left column here, you can see that the spatial distribution of edges is quite different between the two pipelines, and the same is true for the other contrasts as well. So just reaffirming this idea that, if I was doing a case control study and I only did one pipeline, I would do something like this. I would look at these results. I would look at how the edge distribution in the brain. I would look at how they broke down by systems. It's a very common thing to do. And I would sort of draw inferences about what I thought was pathophysiological with respect to schizophrenia in this case, based upon those results. And so this just sort of, I think, says quite plainly that, at least in our hands and in this case, that conclusion is going to be very much dependent on the pipeline that you choose.
Quick question, Linden.
Sure.
So what statistic are you using? Is it the edge weight for functional connectivity or the bold signal per ROI?
This is the edge weight. So this is comparing the functional connectomes using the Andrew Zalewski's network-based statistic approach to assign significant results.
Okay, thank you.
All right, so wrapping up so we can have some discussion. So there's a few challenges. I just thought I would throw a few challenges out there that are in my mind, as well as maybe a few future directions, just hopefully to inspire some discussion and some argument. One of the big problems is, we lack a huge ground truth, right? We lack a ground truth here, and that provides a whole host of issues. It raises a bunch of questions, right? So we don't, how far is too far with regards to mitigating these QCFC relationships, right? I'm making this bold assumption, and others as well, that if we, it's reasonable to drive these benchmarks to zero, but, how far do you go with it? Is that reasonable? How much signal are we throwing out? At what point are we throwing the baby out with the bathwater is unclear, because we lack a ground truth. As a similar sort of knock-on effect of that, we really don't have any idea of, which of the schizophrenia effects are the real ones. I was comparing these two pipelines before and showing you that they're different, but I have no reason to tell you why the ICA aroma ones are more real than the anatomical CompCore ones. And so that remains a big problem. Of course, how we measure cost is also something that we did, I think, a fairly basic or kind of first order version of. This is something I remain sort of dissatisfied with our paper, but but ended up just eating it in the end. You know, we measure it just based upon the number of regressors that were in our model, but whether or not that's a great marker of cost remains unclear to me. And you know, something I mentioned earlier in the talk was, of course, this problem of participant exclusion as it relates to the volume censoring methods. So you know, the more you censor, the less high quality data you have left, and then at some point you make some decision about throwing the participants out of the analysis because you don't have enough good data left, and then you end up with better performance by virtue of just throwing away that data. But you know, maybe that's the approach, right? Maybe that underscores the idea that the only reasonable approach here is to just get the best data, and that if you have to throw out bad data, then you know, maybe that's the only way to go. Who knows? Few future directions. The other big problem for me here is that, you know, these benchmarks we're using are fairly coarse. You know, they just represent sort of proportions of significant effects at a whole brain level. They're very easy to manipulate because they're just based upon p-values, right? I discussed earlier the problems associated with correcting for multiple comparisons. You know, if you're trying to convince a reviewer that your results are sound, it's very easy to make these pipelines look, or make these benchmarks look good, right? To go to zero. And, you know, they're only really kind of meaningful in relative terms within a single data set, right? You can't really meaningfully compare between data sets that easily, again, because they're based upon parametric statistics. So there's power considerations there. Like if you're trying to understand how a single pipeline worked across multiple data sets, if those data sets have very different ends, then you have run into difficulty trying to compare across data sets. The other thing I haven't worked in this space for a while. This is the last paper I did in it. I haven't sort of kept working in it, but you know, every year since I published this paper, I've of course been asked to review lots of papers from NeuroImage doing the same thing, and I've noticed at least a trend for just bigger and bigger and bigger. You know, I find myself reviewing papers that are now comparing 100 different pipelines, or you know, 12 different data sets, and I think that while that's great, and a lot of those papers are still being published, there's a part of me that wants to kind of take a little step back and think about how we improve these benchmarks, how we can find more subtle benchmarks, and you know, and think more about the benchmarks as opposed to just increasing the kitchen sink, if you like. Last, a few resources. So of course we put our code up on GitHub, so you can go and check that out if you like. But really, that was just to reproduce our results. I never really thought of that as like a toolbox or software that I expect people to use. These days, there are far smarter people than me working on far better tools. And so whenever anyone asks me what to do, I always say, go and use fMRI Prep. So I would encourage people to look at the fMRI Prep community, and you know, don't use my code, use their software. It's definitely the better way to go. Then a little bit of a plug on the right-hand side for some follow-up work done by a colleague of mine, Kevin Aquino, who I'm sure would love to come and give a talk to you guys as well. One of the things he did as a follow-up to that work was he did a whole paper sort of thinking about the problem of global signal regression and whether or not we can design algorithms for pulling out the global signal more precisely and trying to sort of dig into that with more subtlety. So you can check that paper out in NeuroImage from a year ago. And that's it. So this is, as I said, work done during my PhD. So the appropriate acknowledgements are my three wonderful advisors, Murat, Alex, and Ben, who are pictured there, as well as Monash University, which is where I was based at the time. Thank you all for listening, and looking forward to probably some heated discussion.
Thank you, Linden. This has been fantastic. Looks like you are my twin brother in presentation style. Beautiful slides and great topics. So I'll leave it up to the audience to ask any questions.
So thank you for the great talk. I just had a question related to kind of how these motion correction steps interact with other steps that may have occurred prior. So I know Dr. Lindquist had published previously that the physiological correction happening before the motion correction can then induce the physiological noise back in the data since they're correlated. So I was wondering how these interact with some of those steps, if you know.
Yeah, that's a great question. It's not something I've looked at comprehensively. The approach we took in this was just to set them, to not vary those decisions. But of course, they will absolutely, all of the massive Gardner forking paths would probably have huge impacts on those benchmarks I showed you. And so that includes all of this decisions leading up to motion correction. In that paper, I'm trying to remember what we did. It was some time ago, but in that paper, we tried to pay very close attention to not reintroducing certain frequencies of signals. It was Martin Lindqvist's work showing that if you don't properly bandpass filter the nuisance regressors, you can reintroduce frequencies that you might have filtered out. So we paid particular attention to avoid falling into that pitfall. But beyond that, I didn't do any sort of systematic looking at whether or not some of those other decisions impacted our results. My pessimistic view is that they probably do. But I haven't kept up on top of the literature to see whether or not someone's been doing that study or not.
So we looked at the Beijing data set too, and yeah, they have really low motion in it, and the low movers are really, really low movers.
Yeah.
But this isn't the kind of thing that most people are really interested in for this kind of motion correction. It's dealing with kids is the bigger problem. So, so maybe the degree of motion is going to, or how much motion there is in your groups is going to be an important part of which pipeline you're going to pick. How you're going to pick those parameters. I remember when we were looking at this, we couldn't, if you separated it by quartiles in the way that Buckner did, our first idea is, well, we could do a motion covariate. Of course, this doesn't work at all because we've separated them by motion. So nothing is left, so it can't work. But if you have two groups that overlap in the amount of motion, then you could potentially compare them with a covariate.
Yeah, absolutely. We had similar troubles with that because I tend towards doing individual differences, and motion, I find in my hands, at least in the data sets I've been working with in psychiatry, that mean frame-wise displacement can sometimes correlate quite strongly with depression symptoms and stuff like that. And so, it's very difficult to know, how much you're just destroying effects you're interested in. And yeah, it's I think a very tough challenge. And I remember having this discussion with someone else who said, oh, well, this is why you should do, participant exclusion-based stuff. So you just retain high quality data, but then you're just excluding the top end of your symptom distribution as well, right? You're just more and more pushing into the subclinical stuff. So, I don't yet have a sort of convincing solution to it. My approach these days is, in all of my papers, to try and is to not overcorrect. I try to be more conservative with the correction, which means, I leave motion contamination in the data, and I've settled on just, given my specific hypothesis, asking the question and putting it in the subs, to what extent the effect I care about is still correlated to motion. And, it's been kind of hit or miss. Sometimes it still is, and that's a problem, and I just wear it as a limitation. Sometimes, fortunately, it's not, and that increases my confidence. But that's generally been my strategy.
So I don't know how fMRI prep handles it, but in AFNI, we don't do global signal regression because it induces correlations, both positive and negative correlations, and we don't want to do that. So we found that these other methods remove it well enough that we can avoid that. There are other methods, I don't know, Daniel Handwerker will be talking about this, but multi-echo is another method that some people requires a different acquisition. handle motion.
Yeah, I've never worked with multi-echo, so I'd be interested to hear what that space is like. I don't know what the current state is.
And another question that Jillian probably has is about prospective motion correction. Have you looked at any of that, or the other people here probably have some experience with that?
We had a talk on this a couple of weeks ago, early in this month, just giving a didactic overview that might help some people. It's a very different space, right? I think we might need a combination of both in the future, but I wanted to go back to that ground truth issue that Linden identified. You know, that was one of my big questions. That we keep coming back to this in various talks in this QC series. You know, it looks like we could use some phantom-based validation for some of these methods. You know, right? I don't know. What do you think, Linden? Would using a dynamic phantom would help validate these methods better?
Yeah, I mean, what's like when you're scanning phantoms, this is not something I have experience with, but like when you're scanning phantoms, what kind of experimental designs can you come up to simulate functional connectivity, right? That then you need to, right? You're trying to, because the idea is like we want to separate our signal from our noise. So what's the experimental design there that creates a reasonable proxy of our signal, I guess?
No, so I guess the reference is there is no functional connectivity, and there is a known amount of motion, and you apply all these methods and they remove just motion only, and not introduce another non-existing bias towards FC, the negative or positive correlations that Daniel was talking about, right? So, I mean, I put a paper in the chat. Basically, they approach it. Their interest is also to try come up with some kind of ground truth signal to provide a more baseline, so to say, you know. Yeah, I think this is one of the big challenges across multiple methods, including motion correction. You know, we need more phantom-based validation. I mean, we didn't even talk about the physiological issues in terms of breathing and other even deeper confounders.
So I want to build off a little bit on that. I'll first say I wasn't planning to talk about multi-echo, but I'm happy to talk more about that offline or later. But on the question of a dynamic phantom, I think it has a place, but it has some of the same issues as anything else, in that you can design your dynamic phantom to be addressed by your motion correction algorithm of choice to a certain extent. So if you hypertune what your phantom does and hypertune your motion correction algorithm, you're getting a little bit too far away from the underlying goal. And I think one of the issues with a lot of these methods, I really like how in your work, you're using a lot of different metrics, but historically, a lot of these metrics kind of were hypertuned to the denoising the method of choice. So I think the classic example is the push for global signal regression as a motion correction fix came along through the Carver plots and metrics that summarize the Carver plot. And by definition, what we're looking, I mean, I think the Carver plot is a good artifact detection tool, but what we're looking at it is the less global signal you have, the better. Yep. So any metric that, you know, effectively summarizes the Carver plot, which I think is one of your QCFC measures, when you remove global signal, the metric's going to get better because that's what the metric is effectively calculating. And this, I think, is where it links into this dynamic phantom question, that we need some sort of control that's less sensitive. And I wonder if there's also a place for task in this, because in task data, we're seeing something that both tasks can be correlated with motion, but we also have one more level of ground truth where we're not saying, you know, when we're just looking at rest at a fundamental level, we're saying, this is what a functional connectivity match should look like. And that's obviously noisy. But if we say this is what a response looks like, we have at least one more metric where we can say, yes, motion correction is helping here. And these denoising metrics should help here. I'm curious if you've tried some of the similar thing on task data or considering it.
Yeah, no, I'm not a task researcher. In fact, I've more and more moved towards structural connectivity, although not that that's without its problems. I do know, so I think like there's a huge line of, I mean, Matthew Glasser does a lot of work in this space, is thinking about how denoising interacts with tasks, as I recall, and I'm trying to remember some of the stuff he's written in his papers. But I mean, yeah, so yeah, I absolutely agree with you, but I don't know, like, what would then, you know, if I dodge the question and reframe it for the value of resting state, are you saying that then you're just saying, well, if I can just get closer to the ground truth, I can assume the resting state's better? Is that the kind of goal?
I mean, motion artifacts should be the same in rest and task. They might affect our results differently, but
Yep.
it's not like the head motion is distorting the data in a unique way in rest. So if your method is removing motion, it should work equally well in task and rest. And I think that's kind of the underlying message, at least of that part of my question comment.
Yeah, but then like the flip side, something I've always worried about and wonder what task-based, you know, sounds like you've got more experience with task-based than me, but then I agree that like the assumption should be that the contamination is like qualitatively similar. But then what about in instances where motion is locked or motion is correlated to task design? You know, how does that impact, like in your hands, how does that impact what you then learn about resting state, right? It's like if people are doing something in a task and there's kind of a signature of motion that links to that design, and then you're trying to sort of move information that you learn from that task-based denoising process to the resting state, do you miss, like, you know, do you effectively have I mean, it's like saying what you were saying before about tuning for a specific benchmark. It's right. If you've tuned motion correction for task, and you have a specific motion signature because of it that's locked to the task design, even if the basic contamination is similar, do you then risk under-correcting the resting state because whatever you've learned about motion correction from task-based is specific to the task design and then does a relatively poor job in the resting state acquisition compared to something just like ICA aroma, which is, well, you know, which is tuned specifically for resting state, for example?
Well, I think that the two things I'd say there are, I mean, you have to be thoughtful about this, but if you're tuning methods, you're still tuning methods. So fundamentally, I mean, I've been looking a little bit at ICA aroma, and nothing in ICA aroma is actually idealized for rest. I mean, they're looking for edge artifacts, which is the exact same on task and rest, and else, which is the same, and they're looking for ICA components correlated with motion. That's the entirety of what's under the hood in ICA aroma. So, I mean, I'm actually planning to run ICA aroma on task and trying it. I don't know if it's been used that way, but there's nothing magical about the method that's specific about rest. And
it was just,
you know,
sorry, you're right. It's just validated in rest, as I recall, like all the benchmarks and the development was done in resting state acquisitions.
But there's also some work, like for task, a motion artifact is going to be immediately task locked, while a hemodynamic response is going to be a bit slower. So that's the thing you can't look at rest, but you can look at with task data. So you actually get a dissociation between your activation pattern, your activation time series, and motion spikes. And a method that corrects for that will actually show you that. So I think there's definitely room to apply these methods. On the flip side, things like global regression utterly fail on task, and that's again worrying. You know, if a motion correction method doesn't work on task data at all, that's concerning because resting data is essentially task data where we don't know the task.
Daniel, quick question. You kept referring to something called hypertuning, you know? Do you want to elaborate more on what you mean by that?
I mean, just essentially a lot of the papers when someone designs a new denoising measure, their metric for success is sort of developed in parallel to what they're trying to do. So
Right.
the example would be you make a metric like a carpet, things based off the carpet plot, which the less global signal you have, the better. And then the metric you're testing is, are things better when we remove the global signal? So it's completely
overfitting. Yeah.
Yeah, the circular.
so it's completely circular. Just to be clear, I think carpet plots, I said this earlier, but carpet plots are great for artifact detection. I think carpet plots and things based on them are really bad for saying the data is better because I've removed, because of things are smoother.
Yeah, I think this goes back to the ground truth problem, so yeah, we need that. So this, Linden, is there a way to, maybe they're already open, to get all this data and different benchmarks coming out of papers that you're reviewing and all in an open challenge that we can probably try design something not so circular and keep it running for like, I don't know, 6 months or something? Make it an actual open benchmark than through peer review from 3 or 4 people,
Yeah, I don't know that. I mean, certainly, I think I put all. You can certainly download my stuff. I put all of that up on Figshare, so that's pretty easy to find. But I don't know. I don't know how available other people's benchmarking stuff is. I don't think it is. I think the code's pretty well available, but I don't know how standardized the sharing of the derivatives has been. But I mean, the benchmarks are trivial, right? I think they're trivial to calculate. The most important thing, the most important thing to get access to from just doing it at scale and challenging people is just the connectomes, or the processed derivatives of the functional data. And so if you wanted to do a really massively multi-data set version of this, then that's the problem to solve, because the QCFC stuff is trivially calculated in a handful of lines, and you can't really get it wrong because there's the Gardner forking path is in the pre-processing, not in the benchmark calculation.
Right. I mean, so this reminds me of a lot of work my postdoc advisor, Stephen Strother, has been doing. I don't know if you came across his work. He analyzes the importance of the pipeline combination. fMRI has these at least 10 steps before we do something useful, such as functional connectivity. And one of the arguments he's been trying to make is that you don't need to do all those pre-processing steps. Depending on what you want to do at the very end, you can optimize what subsets of these 10 steps that you do to do that, right? So what we looked at is we are only fixing and changing one step of that process, like you said, which lies in the bigger context of what many other things you do. I think that's one of the points Daniel and Daniel, both of them are making here, depending on. So I think there is definitely a need for leaving certain data sets and derivatives out there and keeping some kind of, and I guess we'll also have to develop some consensus on what is a good metric for showing that motion correction works. Perhaps we can get Daniel H to lead this development of consensus. Daniel, would you be interested?
Not sure what I'm leading right now, but happy to talk more. So.
Okay, yeah, I think maybe it looks like we may need a few more debates or whatnot,
So this will get going around a few debates.
I'm certainly more interested. I'm certainly very interested in new benchmarks. That was, I think that was the biggest. That's the thing that I still remain dissatisfied with, is just the sort of simplicity and the coarseness of them, and it was something I kind of grappled with in publishing that paper, and because I knew that I was worried, and I was right, that the outcome of that paper would just be that people would use ICA aroma and just say, because Linden said use ICA aroma, and then half the citations on my paper are that. And that always kind of bothered me, because that was never really the point. And in fact, part of the reason why I thought I could add value was because I didn't have a horse in the race. I didn't like, none of the methods were mine. This comes back to Daniel's point around like circularity of optimizing for things that are self-serving. And so like even now, I like sometimes when I'll review papers, people will have included benchmarks in, reporting of benchmarks in the supplementary materials. But again, it's often just we found, one line, we found that the QCFC correlation was 20%. And it's like, that's just such a meaningless number to report in isolation, that it doesn't tell anyone anything about the motion contamination really. And so I think that, one of the things I said in the talk is that people are still publishing this space, which is great, and NeuroImage clearly loves to publish papers in this space. But what I'm seeing more and more of is just doing more and more and more of the same sort of thing, or bigger and bigger and bigger. It's like I need to know what it's like when we do more data sets, and there's value in that, but I think that implicitly people are still trying to hunt for the best method, and I don't know that that's the right question to ask. I don't really care if you use anatomical comp core or if you use ICA Roma. That's not really what concerns me. I just think that the benchmarks need to be refined to a point, or developed to a point, where there's some actual intuition on contamination in isolation of a single study and a single data set and a single choice of pipeline. And I still think that we haven't got there yet. And so, if you're thinking about working groups and developing more ideas, for me, it's the benchmark space is, I think, where it's exciting and where impact could be found.
Are you saying yes to be a member of the task force?
Maybe. I'm not an independent scientist yet, so I have to.
No, I mean, we could have you as a consultant, whatever. So this sounds like the AFNI philosophy about forcing the user to think about what they're doing on the data rather than just taking a pipeline and shove it through the data, if I can put it in a really blunt way, right? So like you said, don't just use ICA Aroma because it turned out to be good for some arbitrary metrics I choose at the time in 2018 or whatever,
Yeah, before that.
Yeah, yeah.
It's hard because if the field, it's like, at the same time, there's that tension, of course, with the field, because it's like the easier you make things, the more likely you are to get people to adhere to it, right? And I think that I don't think that that's necessarily sinister. Like, I mean, I get what you're saying, which is that like there's a problem with making things easy, a push button, and trivial to do without much thought. I agree that there's a problem with doing that, but at the same time, that like I don't think that it's not that surprising that people want to optimize for what the experts say, and so I do think that there's room to kind of meet people halfway, in the sense that experts can sit down, or people who want to develop the benchmarks can hopefully think, try and think hard about what would be benchmarks that actually matter for a paper. And even if they're then easy to do automatically, I don't think that's the end of the world.
Right. That sounds like a small a need for a small review of what are the typical benchmarks for different use cases. I'm not an expert, but that sounds like we need to raise the awareness of the importance of choosing the right metric for the particular type of analyses, rather than just seeing it as a way to remove motion for everything,
Yeah, and it may well, I just want to also say, like, a lot of that work might start existing. Like I did this one paper and I've since not stayed in the space. And this was a paper I did when I was sort of like a bright-eyed, naive PhD student who could have known better on lots of things. So, there's lots that I will not have kept on top of in the last few years since I published that paper. So, maybe people are working on those benchmarks. I'm actually less aware of it than I should be.
Yeah, it looks like we need to get the AFNI people as well as Kevin to talk more. I originally wanted to get both Kevin and you at the same time, but I was worried we don't have enough time to, like, usually these run for a long time. So any other questions? I noticed we have some few new members. Hello, everyone. Thanks for joining. Okay, looks like we gave such a wonderful talk. People are all happy and clear. Thanks again, Linden. This has been great work.
Thank you
Keep
for
in
having me.
touch. Yeah, I will keep in touch. We'll certainly get Kevin and Daphne people in the future. And if there is other important papers, please feel free to email me or send to the group. This series will run for a long time. Because there's so many open questions and whatnot. So we're happy to keep this going. Just feel free to email me or the group anytime. Thanks again, Linden.
No worries. Thank you for having me.